Hi, this is Miss Litton, and this is 7th period review. Say hi.
Hi.
Excellent AP biologists. This is over chapter 3, the chemistry of organic molecules. So this is a review of the whole thing. So the first thing I want to bring up is organic molecules. What does it mean to be organic? You have what in you? Carbon. What's an exception to the rule?
CO2.
CO2, because it's too simple. Yeah, too simple. So we used words like hydrocarbon. What does a hydrocarbon have in it? Hydrogen and carbon. And we looked at a lot of these hydrocarbons and we said we use them as what? Starts with an F. Fuel. We use them as fuel. But then we said when they have these, we're assuming when you look at this picture that they're single bonds, but we talked about how they could enter into double bonds and even triple bonds. And on top of that, we could add in little functional groups that gave those hydrocarbons different properties. And some of those functional groups are polar. Some of them are non-polar. Some of them have what? Charges, yeah, on them. So it can change the chemistry of that particular organic molecule. There are some up here that we became really familiar with. If you see this right here where it says carboxylic acid, C double bond OH. Where did we see that? That's an important part of one of your bricks. Amino acids, good. Okay, and we talked about an amino group on the other side, a central carbon, and then what was the other thing that distinguished an amino acid?
R group.
R groups. How many different R groups?
20.
20. So we were looking at things like fatty acids, and we looked at nucleotides, and we were studying all those different functional groups, and you have a summary of some of the different ones. But one thing that's important is understand if something's hydrophobic or hydrophilic. And we know hydro means what? Water, and philic means love. So if it's water loving, it means it can interact with water. What do you need to be in order to interact with water?
Polar.
Or charged, yeah. If you're polar or charged, then water, who has the hydrogen ion, the hydrogen portion of the water is partially a water molecule, H2O. The hydrogen part of the water molecule is partially positive, and the oxygen portion is partially negative. So the water will orient itself around your charged or polar molecules in relationship to the whatever they have on them, their charge. Okay, we talked also about isomers. Isomers, you could have C6H12O6, but the arrangement of those atoms, where they might have their single or their double bond, that part can vary. So that is an isomer. You have the same molecular formula, but different arrangements. Then the next item we got into was discussing monomers and polymers. And this was the bulk of this chapter. Okay? And what we realized is use monomers to form polymers. And you can take your polymers and you can go backwards back into monomers as well. There is a key reaction that we do again and again and again. Whenever we have monomers and we want to build a polymer, you should know the name of that key reaction. And there's one that goes the other direction, taking a polymer back to a monomer. In your own head, come up with the names of those reactions. Don't say it out loud. Don't whisper it to your bio buddy. Just see if you have it. And the one on the left actually has 2 names. Can you think what the 2 names are? Okay, everybody got a chance to do it? Okay, so when I want to go from monomers to polymers, what am I going to use? Dehydration synthesis. Yes. And what's another name for it? Condensation synthesis. Perfect. And whether I'm getting dehydrated, which means, thank you, sweetheart. Whether I'm getting dehydrated, what am I losing getting dehydrated? Water. Okay, so I'm going to need, these molecules are stable all by themselves. So I have to get them, they have to be needy so I can join them together. So from one, I might take away an OH, and from another one, I might take away an H, and then together that will form what? Water. But now they will join together as a result of that. I can hook them together. Dehydration synthesis. Another name for it, condensation, is because you're talking about both ends of the reactant and the product side. I'm either going to take away, dehydrate them, and remove a water, or on the product side, I produce condensation synthesis. I produced a water molecule. All right with that? If I want to go backwards to do, take my polymers and make a monomer, what is the reaction I'm going to do? Hydrolysis. And we're going to remember that because hydro, water, and lysis. Now the next thing you need to have in your mind is we discussed that there were 1, 2, 3, 4 important organic molecules. In your mind right now, can you come up with all 4? Don't whisper them to your bio buddy because they're trying to do it themselves. Think in your mind, what are the 4 important organic molecules just in your head? Again, we're not whispering them because you're trying to think them in your head. Okay, what's the first one we discussed?
Carbohydrates.
Perfect. We talked about carbohydrates, so carbs. Okay? What is the second one we discussed?
Lipids.
Lipids. What is the third one we discussed? Proteins. Okay, what is the fourth one we discussed? Nucleic acids. Okay, now your job, my friend, in your own head, not being whispered to by a bio buddy, in your own head, without looking at a document, come up with the monomers for all 4 of those, just in your head. Give it a second. Could I come up with that? Could I write it down? Apparently my phone's on. All right, you ready?
Yeah.
So you could tell me that the monomer for carbs are what? Sugars. Beautiful. How could I always recognize a formula for a carbohydrate? CH2O, carbohydrate, right? Okay. Now lipids. Lipids are a little bit different, right? Because lipids, we have 2 big categories. What are our 2 big categories?
Fatty acids and
Fats, yes, or fatty acids, and what?
glycerol.
No, steroids. Right? So on fats, we're thinking, okay, what makes up a fat? We could talk about glycerol and fatty acid chains, right? But then we could talk about waxes. We can look at a modification of that. We're talking about phospholipids. Those would all be related to that. Steroids look totally different from fats. Steroids are made out of what? 4 carbon rings. Good. All right, good. Okay, now let's move on. Proteins. And when I say these things, are you imagining their structure in your head? Because imagine that on your quiz tomorrow, I'm going to have these structures and I'm going to say, what monomer is this? Or what could this build? Right? Imagine that. Imagine those dragons, because it will happen. Okay, what are proteins made out of? Amino acids. Are you imagining the structure of an amino acid? Beautiful. Central what? Carbon. What's right up above it? Hydrogen. Over here. Carboxyl. What's over here? Amino. What's here? R. Perfect. Okay. Then the fourth one. What are nucleic acids built out of? Nucleotides. And then you go, but there's more. Because a nucleotide is made out of 3 things, and I can tell you what that is. What is it? Phosphate group, nitrogenous base, and a 5-carbon sugar. And we know those sugars can vary, and those bases can vary too, right? Those are all in our head. You're not stressed out about it. Okay, so all of these polymers are built the same way. Through what process? Dehydration synthesis. And all of them can be broken the same way by hydrolysis. Could they have different functions, these polymers? Okay, so that's the big ticket. Different organic molecules are built in the same way and broken down the same way, but they serve different functions in our cells. So let's just hit a few of those as a reminder. We did this. Oh, I'm getting cartwheels. I don't need that. You already did that. Ooh, I know what I want to talk about. What's an important requirement usually for these reactions, whether we're building or breaking? You need a something that starts with an E. Enzyme. Good. And enzymes are named after their, you're so close, substrate. Right? Right? So cute. How did you all get though? Did I say
No, I think
somebody starts. Yeah. I'm like, it is. What digests sucrose? Sucrase. There you go. Okay. Very nice. All right. So we start with carbohydrates, and I'm just going to do a quick review. We want to be able to recognize it. So if on a quiz, I draw something that looks like this. Maybe I put an O there, maybe I don't. You want to be able to recognize that looks like a sugar molecule, and that will build carbohydrates. Okay? Get that in your head, what it looks like. That shape right there. It could have some things on it like this, but I know and I can recognize sugar, and that will build carbohydrates. Look at your bio buddy and go, did you know that? You comfortable with that? So, come back to me. That is a monosaccharide, but it could, two of them could join together through dehydration synthesis, and I could form a disaccharide. They all have glucose in common, right? They all have glucose in common. I'm not going to see if you memorized all the disaccharides tomorrow, so if you're going to spend time memorizing that, set it aside. It's good to have in your wheelhouse, but I won't have that, I promise you, in your quiz tomorrow. But I bet you could tell me all of those. Sucrose is glucose and fructose. What is maltose? Glucose and glucose. What is lactose? Glucose and galactose. Okay? Things like maltose and sucrose and lactose could be used as what? Energy. But if I made some polysaccharides, I could store them more efficiently as a large polymer. How do you and I store large polymers of polysaccharides? As something. Glycogen. And we store it in our livers. Glycogen is in the liver. Say it. Glycogen is in the liver. Tell me one more time. Glycogen is in the liver. And glycogen gives us what? Energy. But now I'm a plant. Plants don't store their carbohydrates as glycogen. They store it as something else. We eat it all the time. It's called starch. Perfect. Say it again. How do plants store energy? Starch. But they could use the same molecule. They could use glucose. They could arrange it differently and make it very, very strong to build their cell walls. Now it's doing something structural. So let's build this same brick, but make a what out of it? Wall, and it's going to be made out of what? What is it called? Cellulose. Can you and I digest cellulose? No, we cannot. We don't have the enzyme to digest cellulose. But animals could also use it for structure. What could they build with it? Exoskeleton. Like what? Made out of what? Chitin. Good, chitin. Chitin. You're full of chitin. Okay. Guys, it's 2:49. I still have other things to review, and I want to help you because you're important. Okay? So polysaccharides are not soluble in water, too large to pass through membrane. So we already know energy storage. Tell me energy storage too, right now. Glycogen, starch. Structural molecules, give me some. Cellulose. Chitin, say it. Chitin. Okay, next. You have a little summary of this. Right? Did we go over all of that? Yes, we are so excited. So now we're going to talk about fats. Now we have many different categories. Lipids. We have many different categories of lipids. One is fats. Fats are glycerol, right? with these long fatty acid chains. Glycerol and fatty acid chains to build a fat. We know those fatty acid chains can be in two categories. What are the two categories? Saturated and unsaturated. Saturated fats means they're saturated with what? Hydrogens. This makes me think that there's a highly suggested reading and thinking that you want to answer so you can get it right on your quiz. Yes? Yes. Saturated with hydrogen. Since it's so saturated, the chains are all straight, no double bonds. It stacks up and it'll be a what at room temperature? So, what kind of fats tend to be solid at room temperature? Animal fats. Whereas if we have, where could we get unsaturated fats? Plants. And it tends to be liquid. What are some plant fats that we use all the time? Olive oil, canola oil, right? Any kind of oil is liquid, okay, except for palm oil. All right, so fats and oils, glycerol and fatty acid chains. Now let's do a modification of it. Let's remove one of these fatty acid chains. The glycerol is still 3 carbon, and let's add on a phosphate group. Now it's called a phospholipid. Phospholipids can be found where? Cell membranes. Cell membranes. Okay? Cell membranes. You have 2 layers of phospholipids in your cell membranes. Why they work so well? The long fatty acid chains are hydrophobic, but the phosphate groups are hydrophilic. So we put them on the outside towards the liquid medium of the cell or towards the inside towards the cytosol. We're going to learn all about them in chapter 5 on Thursday because we're not taking our test on Thursday. We're learning about chapter 5 on Thursday, right? Okay. Steroids. Steroids look totally different. If I showed you a steroid, you would see something that looks like this, though I didn't do a good job here at all. What am I trying to draw for you there? 4 carbon rings. You know how you're going to remember that? Somebody might win a lot of rings in sports because of all the what? Steroids. So they have all their carbon rings. They're like, look at my carbon rings. I'm really good because I took a lot of steroids. All right. And then I'm literally just throwing waxes in here. This would be a modification of just looking at, you would have a long chain fatty acid with a long chain alcohol. I'm just throwing in waxes fit under this umbrella, because waxes protect what? Leaves and animal fur. Good. It might be in your ears. Good. So that is all underneath lipids. So this should look familiar to you. Guys, hold your smiling poo together. Stay focused so we get through this, right? Okay. Okay, I forgot. Okay. So then we know about this already. Okay, so I'm just going through that quickly. Why do we need fats? Energy. What else? Cell membranes. Insulation. What else? Tastes good. We might eat them. Waxes, right? All those things we listed. Those could be their functions. All right. Moving on. Oh, don't eat too much, otherwise you'll clog your arteries. Yes? And here was the review on lipids. You understand everything on there? Hormones coming from steroids, right? Okay. Next, proteins. You want to know proteins are everywhere. Okay? They have functions all throughout your body. You need to be able to recognize the structure of an amino acid. Amino acids build proteins, and we already went over that. Amino group, hydrogen group, carboxyl group, R group. 20 different R groups, and they can have different properties. When we want to join them together, it's got a special name. We're still doing dehydration synthesis, but the bond between the amino acids is called a what? Peptide bond. Now, we need to talk, remember primary, secondary, tertiary, quaternary structure?
Yes.
Let's run through that. The basic primal structure of any protein is the sequence of what? I'm hearing it. Sequence of what? Amino acids. Boom. Sequence of, it's primal, primary. Now level up 1. The second part has to do, secondary structure, has to do with the either end. You have a carboxyl group and you have an amino group. And they have charges on them, so they can be attracted to one another. So they'll go into one of two conformations. They'll either be a alpha helix or a beta pleated sheet. Does that have anything to do with the R groups? No, nothing. Primary structure, sequence of amino acids. Secondary structure, either a what? Alpha helix or a, and that's just because of either end. It's these two ends we're worried about, right? This end and this end. 1, 2, secondary structure. Tertiary structure is when we what it? Fold the secondary structure. What is that dependent upon? R groups. That is dependent upon your R groups. Because hydrophilics are going to want to get together and hydrophobics are going to want to get together. Okay, and then quaternary structure is when you have more than what?
2.
Yeah, more than one chain, and they're connected together into a globular protein. We talked about the importance of, there are other proteins that actually fold the proteins to make sure that they are folded correctly. And we also talked about diseases that you could have as a result of misfolded proteins, and those are called prions. And then this is where we are. Proteins, here's the amino acid, muscles. The only thing I wish I would have added to this diagram is this right here, and I think we talked about this in class. Primary, secondary, tertiary, quaternary structure. All right. Last but not least, we talked about nucleic acids. Nucleic acids are built out of what? Tell me. Nucleotides. Great. And nucleotides have 3 components. They have a nitrogenous base, a sugar, and a phosphate. Now, could you recognize, if I gave you a nucleotide, could you say, that is found in RNA, whereas this is found in DNA? What's something they both have in common? Okay, they could both have adenine. What else? No. No. They do both have 5 carbon sugars, but not the same one. DNA has the sugar what? Deoxyribose. Remember? DNA has, that's why it's called DNA, deoxyribose. Okay, and RNA has the sugar ribose. Remember? It's crazy. Cray. All right. So DNA and RNA, it starts with a D or it starts with an R. So DNA has the sugar, deoxyribose. I always start with the sugar. Eat dessert first. Ribose, R, RNA. Okay? What? Yes, eat sugar. I sure diabetic. Okay. What else do they have in common? What do they have the same of? Okay, they both have guanine, they both have adenine, they both have cytosine, they both have phosphate. The only thing that's different is DNA, besides deoxyribose, what base does it have? What is it? Thymine. And RNA has the sugar ribose and uracil. Are you kidding me? Ribose, uracil. Ribose, uracil. Are you kidding me? Uracil. Okay?
Are you? Oh. Okay.
So the other thing we learned was that nitrogenous bases are either purines or pyrimidines. And remember we said poor primitive pyrimidines. You only have 1 ring. Whereas purines have 2 rings. Purines have 2 rings. Which 2 are purines? A and G. A and G are purines. And we remember that because AG, and my stupid way of remembering it is agriculture, pure earth world. You can remember pure silver, good for you. Okay? We are the world. Okay, here's structure of DNA. We can see nitrogenous base, sugar, phosphate. Now, you're going to want to be real comfortable with that. Like, you could almost recreate a DNA molecule eventually. Okay, once I get into that chapter. But for right now, you just want to be familiar with it. And then one other nucleic, here's RNA. single-stranded, has uracil instead of thymine, and it has ribose instead of deoxyribose. And then last but not least is ATP provides energy to the cell, and that is an adenine, a ribose, and phosphate, phosphate, phosphate. So it's a modification, right? It's a nucleotide. It just has bonus phosphates in it. And then we finished out our four groups here. Fourth group, and we drew a nucleotide, and then DNA is our genetic code, RNA is involved in protein synthesis, and ATP is for energy. Okay, you are going to log on to Class Lab. We are going to take a monster quiz. We're going to divide into teams.
What should our names be?
Make your team be your first name. Sign in with your first name.
Wait, how do you choose? Wait, where?
Class lab.
First name. Just put in your first name. No, just your first name.
Wait, is it Smart Lab?
Yeah, it's Smart Lab. Same as Class Lab. Same as always.
I was like,
I think I might.
oh, I know. What is this?
Why can't I hear any sound? Okay, are you in? I have no idea why I'm not getting sound. I'm very sad because you would like this. We were going to divide into teams. You ready? We'll be 6 teams.
What? 6 teams of 5? 6?
Yes. Okay. Find who's in your team. Okay, you ready? You ready? You're going to get individual questions. Ready? Go.
Oh my god.
Oh, Mega Melters, you're doing very well. It'll make you go back if you miss it. Good job. Good job, Icebergers. Fire divers, you're really struggling.
Hey, you know what? I'm doing my best, okay? Oh, you switch up the order of student voice. I know we might go together. I know it has to be like really good time.
Box busters. Oh, Mega Melters.
Fight me already. This class.
Oh, I messed up again. Why am I come on?
Icebergers, go! Frostbiters, go!
Look, it's in the
Oh, so close! Icebergers! Mega Melters, you're out! Almost, Mega Melters! Mega Melters, first place!
Wow, okay.
I didn't know I was doing.
Barrel Busters! Oh! Oh! Frostbiters! There we go. right, let's check here. Oh, look. Finishing time. Okay, here we go. Hydrocarbon contains carbon and hydrogen. That's true. Almost all of you got that. The hydrolysis reaction brings two monomers together. True or false? False. Which of the following are energy storage? This tells you how many of the class got it right the first time. You want it to be green. Starch and glycogen. Did you get that? Yeah. Okay. Some of you said DNA and RNA. Phospholipids make up cell membranes. True. Amino acids are the monomers for nucleic acids. False. Almost 25% of you missed that. Saturated fats do not have double bonds between the carbons. That is true. Okay, this one we missed the most. The secondary structure of proteins depends upon the R groups of the amino acids, charged, polar, and nonpolar. True or false? False. Secondary structure is either one of two things. What? Alpha helix and beta pleated sheet. Is this true about the R groups, that they are this? Yes. But the R groups don't determine the secondary structure. What do they determine? Tertiary. Uracil is found in RNA only. Adenine is found in both, and ATP is the energy source. Good job. And if you would like, I have one more. I don't know if you got a chance to do these in class or not, so I'll throw them out there. Here's some other questions for you to answer. Go ahead. I'm going to pause this for a second. The activity has ended. Okay, I'm going to go ahead and stop it because I think some people have left the review already and want to give you a chance to go over these. Most of you did very well. Nucleic acids are built out of nucleotides. Only one of you said amino acids. Amino acids build proteins. Amino acids build proteins. And by the way, I'm recording all of this for YouTube, so you have all these to look at again, okay? 4 rings of carbon build steroids, which is a type of lipid. Number 4, sugars like glucose build carbohydrates. A couple people picked lipids and fats. No. What do you use to build lipids? And specifically fats. Fatty acids and glycerol. Good. Number 5, nucleotides build nucleic acids. Good job. Oh. So let me just finish up. There's just 3 questions here, and I want you to have them if you want them. When you do dehydration synthesis, first thing in your mind, are you building or breaking? Building. If you're doing dehydration synthesis, you're building. So this is like your strategy of how to do things. Read the question and you think, okay, dehydration synthesis, it's about building. Okay, I must be taking monomers and making what? Polymers. Right? So when you look at it, amino acids you form of proteins. Do amino acids build proteins? Yes, that's a monomer. This is your monomer, and this is your polymer. Amino acids, and you form a water molecule, will I? Yes, because in the process, I've got to remove a water molecule to make them want to be together. Fatty acids and glycerol will form a nucleic acid? No. What would I form? Lipids, specifically a fat. Polymers, you form monomers? No, it's backwards. Mono to poly. Nucleotides to form? Yes, that's a monomer, that's the polymer. 2 freshmen? No.
My bad.
Okay, and then this one, when you do hydrolysis, are you building or breaking when you do hydrolysis? You're breaking. Okay, so when you read, guys, try not to talk when I'm talking so I can finish. When you do hydrolysis, you want to think break, and you want to think poly to what? Mono. So when you do the hydrolysis to a fat, do you make sugars? No. Do you make fatty acids? Yes. Do you make nucleic acids? No. Do you make glycerol? Yes, because these two together will make a fat. Water, do I produce water? No, I require water in order to do it, and not oil. Okay. And last, Which of the following is a DNA nucleotide? Why am I ruling out A? Because of the uracil. Why am I ruling out B? Ribose. Why am I ruling out C? Why am I ruling out D? Yes. All right, my preciouses, love you very much. Have good evenings. Make good choices. Always make good choices.